So I was asked to sort of introduce myself for the first couple minutes. My group works at the intersection between several fields, including signal processing and machine learning, neuroscience, and biomedical engineering. And so, what do these fields have in common here, right? And so, traditionally, not much, right? But over the last few decades, in neuroscience, people have developed methods to monitor the brain's activity and structure in ways that were not previously possible. And this has led to large and noisy data sets, right, where people trained in biology might not have the quantitative skills to analyze them. And so there's been a lot of fruitful interaction between people trained in biology and people trained in quantitative fields like engineering, computer science, statistics, et cetera. And so why are we doing all this? Well, ultimately, what we hope to do is to help humans, right? And so we can take the knowledge that we develop here to develop devices that can help people. Okay. All right, so how did I end up sort of here in this research? So I was an undergraduate in electrical engineering and computer sciences at UC Berkeley. And there I didn't take a single biology class. I didn't really know what a neuron was. And it was when I started my PhD that I was looking for a research group. And so I was fortunate enough to join the group that I ended up doing my PhD in, which is Krishna Shenoy's group at Stanford. And there I did monkey experiments, and I also developed statistical algorithms for analyzing neural population activity. And this was a group that included both neuroscientists and engineers. And so it was very interesting to, at the beginning, where we didn't speak each other's language. But over the course of many months and years, we learned from each other, and we learned to speak each other's language, which was very helpful. Okay, and then afterwards, I did a postdoc jointly at Stanford, also with Krishna Shenoy, and University College London with Manish Sahani. And there, it was a collaboration between the two groups, where the experiments were done at Stanford, and the computational work was done with Manish. Okay, and then I started as faculty at Carnegie Mellon about 8 years ago now. All right. Okay. So to sort of motivate what we're going to be talking about today, there are lots of ways to monitor the brain's activity, right? So this ranges from methods like EEG to fMRI to multi-electrode arrays that you implant right into the brain, right, where each of these shafts, right, you can record from 1 to a small number of neurons at the same time. And so if you have 100 of these shafts, right, you can record on the order of 100 neurons. Also, optical imaging is a very promising technology, right, whereby you inject a sort of dye into these cells, right? So each of these blobs is a neuron, right? So I don't have the video here, but you could play this as a video, and you'll see these blobs become brighter and darker, right? And that's correlated with their activity, right? So in one of these images, you can get the activity of many tens, if not hundreds, of neurons activity all at the same time. Okay, so in all of these different sorts of recording modalities, what we have is multi-channel recordings that are noisy, right? And then a question is, what can you do with these recordings if you want to study some scientific question about the brain with them, right? So the traditional way of going about things is to analyze the activity of one neuron at a time, or one channel at a time, right? And that's all fine and good, but you might ask, is there something more that we can get out of these types of recordings, right? And so it turns out that if you're looking at the activity of two neurons or two channels, right, the way in which the activity on those two channels co-varies is also important. Right? Okay, now if you have three neurons or three channels, right, looking at the covariation pattern among all three neurons or all three channels is also important. Right? And you can scale up to, say, 100 neurons or 100 channels. Right? Looking at how they all co-vary, right, might tell you something beyond looking at how each one of the channels or each one of the neurons activity changes. Okay, so that's sort of the central topic, right, of today. All right, so if we record, okay, we stick one of these electrode arrays into the brain. Okay, in this case, I'm going to give you an example from the motor cortex. Okay, you get recordings that look like this. Okay, so the details of the task that this monkey is doing is not important for this. Just know that this is recordings while the monkey is doing something with its arm. Right, it's moving its arm. So what we can do is to record from tens of neurons at the same time, where each row here, it corresponds to one neuron, and the horizontal axis is time. Right? So each one of these tick marks denotes the time at which that neuron gave off a spike. Okay. And so what you see here is a recording from about 2 to 3 seconds. All right? And so in a typical experiment, we have a couple thousand of these trials. Right? This would be one experimental trial. So we have 1 to 2,000 of these. Right? So you can imagine what the challenge is, right? If you want to just stare at these pictures to try to make out what these neurons are doing. Right? So it'd be nice to come up with some sort of quantitative way of capturing or extracting what's important from this activity. Right. So I forgot to mention at the beginning that I really like to interact. So feel free to raise your hand to ask questions. Yeah?
So in terms of ordering the neurons, is it?
They're not ordered here. It's random.
Oh, so like there's no information about like which neuron is detected by what kind of chunk, and then the neurons that detected to the next chunk is.
You mean the neighboring electrode? Yeah, there might be structure there. Yeah, yeah. So in these types of recordings in the motor cortex, we haven't seen a ton of sort of structure there, although people have reported sort of waves passing over an array. So there's probably structure. And depending on the brain area in which you're recording in, there may be more or less of that kind of topographic structure. Yeah, absolutely. But the way these are plotted here is random ordering. Yeah. Mm-hmm.
You mentioned that there are like 1,000 trials.
Yeah.
Keep doing this thing 1,000 times.
Oh, yeah. Yeah. So each one of these is just one arm reach.
But doesn't he get bored of doing this 1,000 times? And does the activity correlates with the time that the trial is going on?
Yeah. So animals are happy to do this for hundreds, if not 1 to 2,000 trials. So in terms of the neural activity during the course of the day, so in general, we do see changes, sort of slow changes over the course of the day. And there could be a number of factors because of that. So one is recording instability, right? And so this includes both the way, so these arrays, they're in the brain, and the brain is moving, right? Because beats with the heart, right? And so it's possible for these neurons to slightly move relative to the electrode tip. And so you can get, say, better recordings at the beginning of the day and maybe slightly less good recordings at the end of the day, or the opposite. Another possibility is that the animal's arm is getting tired, right? And so at the end of the day, sort of different sets of muscle contractions are being driven to make approximately the same arm movement. Right, and so yeah, I mean, I think teasing these things apart is interesting, right? Because if what you want to study is how things change over the day, then you need to explain. You need to come up with all these different factors, right, and use them to explain what's going on. Okay. So one possible way of studying these types of large-scale neural recordings is sort of a broad class of methods we call dimensionality reduction. And so what is dimensionality reduction? Okay, so I'm going to just sort of introduce this idea sort of from a high-level way, and then we'll get down more into specifics. Okay. So the basic rationale is the following. Okay, so we know that neurons connect up in networks, right? So it's not the case that each neuron acts independently of other neurons. Okay. And so because, say, one neuron, when it's positively interacting with another neuron, right? Then if one neuron's activity goes up, then the other neuron's activity also goes up, right? So these are sort of network level interactions. And so because of these sorts of interactions, right, the brain has fewer degrees of freedom at its disposal than the number of neurons, right, that make up these networks, right? So if two neurons are perfectly correlated with each other, right, there's only 1 degree of freedom in their activity, right? Okay. And so the analogy I'd like to give, right, is a video, okay, where each pixel in the video is a neuron, okay, and the pixel intensity is the level of activity of the neuron. Okay. So here, this video has about 1 million pixels. Okay, so I'm going to play this video. Okay, and what this is basically a ball that's sort of rolling from the upper right sort of to the center of the screen. Okay. So here, right, so each pixel, right, is an analogy for a neuron, right? And so there are about 1 million pixels here. Do we need 1 million variables to describe what's going on in this video? No, right? So if this were a ball rolling on a table, right, you could describe it with 4-ish variables, right? So if you know its position and its momentum, right, then you fully captured everything about this physical system, right? You can predict what's going to go on, what's going to happen in the next frame, right? Okay. So even though the observations here are 1 million dimensional, we only need really 4 variables to capture what's going on. Okay. But when we're studying the brain, it's not so clean, right? So first of all, we usually can't record from 1 million neurons, right? Second of all, the data are noisy, right? And then third of all, neurons aren't always organized in, a perfect sort of two-dimensional grid. Okay, so to make this more realistic, okay, what I did here was to subsample the pixels. Okay, so now we have a 10 by 10 grid of pixels, right? This is like recording using an electrode array, right? So you have 100 different pixels here. So here's the video for that. Oops. Okay. But then in experiments, it's still not this good, right? So we can have noise, right? Something like this, right? Okay, so the challenge is, okay, you have a recording like this, right? Can you make out the fact that there is a ball rolling from the upper right to the center of the screen, right? That's sort of the challenge that we're faced with, right, in the neuroscience context, right? We have high-dimensional, noisy recordings. Can we summarize these recordings using a smaller number of variables? Okay. All right. All right. So the sequence of frames is fully specified by Newton's laws, right? So one could ask, right, whether if you're analyzing neural activity this way, right, are there some Newton's-like laws that govern, right, how the neural activity changes over time? Right. Okay. So the key challenge here is, can we identify a low-dimensional set of variables, okay, that describe these high-dimensional observations? Right? And this is the concept of dimensionality reduction. Okay. So now back to the neuroscience context, right? So let's say that we have 3 neurons, right? Here are their spikes over time. Okay, so what we can do is to define this space on the right where each axis corresponds to the level of activity of one neuron. Okay, and so what we can do is to take a window in time, right, and to count the number of spikes that each neuron gives off. Okay, that corresponds to a point in this space. Right, and then we can move this time bin along, and that traces out, right, a trajectory of points in this space on the right. Okay. So one thing we could do, right, is to analyze this neural activity as is. Okay? But as we mentioned, we typically think of these neurons as having some activity, okay, that's corrupted by noise, and that's what we sort of observe or record, right? And so what we want to do is to do some denoising, okay, on this raw neural activity, and we'll talk about the different assumptions that we could use to do this denoising in just a moment. Okay. Yep.
When you say activity, you mean like firing rates?
Yeah. Yeah. Exactly. Right. So 2 ways in which we could try to remove the noise here is one, right? We can ask whether these points, right, tend to reside in some lower dimensional space within this high dimensional 3-dimensional space. Okay, so in this cartoon here, these points sort of lie close to this plane, right? So we might say, oh, well, what gave rise to what we observed, right, was activity actually taking place within this plane, okay? But the reason why each data point was outside the plane was because of noise. Yep?
So at the point where you go from spike trains to firing rates, do you abandon like phase relationships and correlations between?
Yeah, so within each bin, right, that information would be lost, right? Because, because we're taking the counts. So there's a trade-off there sort of in the bin width, right? So if we choose smaller bins, then we have more time resolution, right? But then the counts are usually 0, and very infrequently they're 1, right? And then, but if you use larger bins, then you tend to get sort of, you can get 0, 1, 2, 3, 4 counts within the same bin. Yeah, right. So the sorts of methods that we're going to consider today tend to work better when you're able to see counts like 0, 1, 2, 3, 4, right? If it's all sort of zeros and every now and then it's 1, we need sort of adapted versions of these methods that can deal with sort of point process spike timing data. And people are working on that. So there have been a number of recent publications that have proposed methods like that. But those also tend to be computationally more expensive because they're really dealing at a fine time scale, right? So we can do it. Yeah. Yep.
In this way, you are just eliminating the noise orthogonal to the manifold, right? Seems.
Yeah, you can also eliminate noise within the plane. Yeah, we'll talk more about it. It depends on sort of the assumptions that you put in. And different methods make different assumptions about what the noise is. Okay, so one way of sort of denoising the data, as we sort of talked about, was saying that the neural activity resides in this low-dimensional space. Okay. Another way of removing noise is the assumption that the neural activity changes smoothly over time, right? So if the neurons that you're recording from, if it's a reasonable assumption that firing rates change slowly over time, right, then then ask whether there's a smooth transition, right, in this space. Right? The idea here is that if neural activity jumps around in this space, right, that's just like saying, okay, a neuron's activity could be 2 spikes per second one moment, and then 100 spikes per second at the next moment, and then 50 spikes per second at the next moment. Right? Maybe some neurons can do that in some brain areas. But for most brain areas that I know of, firing rates change smoothly over time. Okay, so oftentimes this is a reasonable assumption. Okay, so if you do both of those, right, then what you'll get is a smooth trajectory within this lower dimensional space, right? And then you can both visualize neural activity within this lower dimensional space, as well as try to address scientific questions here in a more sort of concise way, right? Using a smaller number of variables than the original sort of 100 variables. Okay, all right. So there's been a lot of recent work using dimensionality reduction, okay, to address scientific questions, right? So Jakob's group and Christian's group have done a lot of great work in dimensionality reduction, okay? But I'm just going to highlight a few sort of scientific studies, sort of organized here by sort of neural system, right? So people have used dimensionality reduction to study decision making, learning, motor control, olfaction, working memory, visual attention, audition, and more. And for review, John Cunningham and I recently wrote this review that sort of talks about a bunch of these. Okay. So what are sort of the scientific motivations for using dimensionality reduction? Okay, so one main motivation is if you want to characterize what the brain is doing on a single trial basis, right? And so what I mean by this is sort of traditional ways of analyzing neural activity has been in the form of things like tuning curves and spike histograms, right? So both of those are based on averaging the activity across many trials, right? And depending on the scientific question that you're studying, right, what might actually be going on, right, in the task, for example, if you're doing some sort of cognitive task or decision-making task, right, you might ask, right, whether what's going on in the brain is exactly the same on every single trial, right? So for example, right, if you present, let's say I show you this integral, right, and I say, okay, solve this integral, and then I have you solve it 10 different times, probably sometimes you're going to solve it really fast, and sometimes it will take you a little longer to solve it, right? And so how can we capture the fact that the neural activity is proceeding differently on different trials? Okay? And so one way of doing this is by leveraging the activity across many neurons, right, and asking how the neural activity is changing on a moment-by-moment basis, right, across the population. Okay? And so to do the denoising, right, so using for tuning curves and spike histograms, the way you denoise, right, is to average across trials. Right here, right, if you're recording from many neurons at the same time, you could get denoising by looking across many neurons, right, within the same time bin. Right? So that is sort of the first motivation here. Right? If we want to analyze what's going on in the brain on a moment-by-moment basis without trial averaging, right, this is one way to do it. So we do have time courses within individual trials, right? But if we want to get down to sort of the millisecond time scale, right, then sort of the basic methods that I'm going to be talking about today are not the best for that. We need sort of to adapt them to sort of have point processes on the output end. And there's been recent work on that. So yeah. Yeah. Okay. So the second motivation is hypotheses about population activity structure. Okay, so what I mean by this is you might be asking a scientific question, okay, where the scientific question is fundamentally about how the activity of different neurons covaries, right? And so in order to address that question, right, you need a way to characterize how the activity of different neurons covaries, right? And so without getting into the details of these studies, so we could get into the details and talk about this later, but I don't sort of really want to get into it now. But I think if you take a look at some of these papers, you'll see that at the very beginning of the paper, like the hypothesis is about, right, how neural activity covaries across neurons, right? And so for that, we need a way of quantifying it. Okay, and then the third motivation is exploratory analyses of large data sets. Okay, so one of the things that we've used dimensionality reduction a lot for is whenever we do an experiment, right, the first thing we do when we want to triage the data set to figure out whether the neurons are recorded stably over the course of the entire session, whether there was any odd trials, right, we usually just feed it into one of these standard dimensionality reduction algorithms, and then we see whether the thing that pops out is what we would intuitively expect. Okay, so for example, if the animal were reaching to 8 different targets, right, then what we would expect is when we look at the neural activity, right, that there would be 8 clusters of trials, right? If we don't see that, then something is wrong, right? Or if there are a few trials that are sort of unlike all the other ones, we would immediately see that as well. So that's the third motivation. Okay, so now I just want to give you sort of a high-level overview of different sort of basic dimensionality reduction methods that are available. Okay, and I'm happy to go into the details on any or all of these as desired on the board. Okay, so the most basic dimensionality reduction method is principal components analysis, right? And this method has been around for over 100 years. And so the idea behind PCA is that we want to capture as much of the variance in the activity as possible. Okay. So in terms of using PCA for analyzing neural activity, okay, we typically use it for trial averaged neural activity. So for example, if we have the spike histogram, all right, for all the neurons for each condition, right, then PCA can be applied, okay, to analyze these PSTHs of the entire population. Okay. So the reason why I would advocate using PCA for trial average analyses is that PCA has no concept of noise. Okay, it doesn't. There's no explicit noise model. Okay, so it doesn't do a great job at denoising. So you sort of need to denoise in advance, and that's what creating the spike histogram is doing. Okay. If you want to do single trial analyses, right, rather than using PCA, right, I would suggest using factor analysis. Okay, and the reason why is that factor analysis has an explicit noise model. Okay, whether the noise model is good or not, right, is something to be evaluated empirically, right? And if the Gaussian noise model that factor analysis has is not the most appropriate for what you want, right, you can switch that out, right? And we can talk about how to do that. Okay. So factor analysis is good for single trial analyses, okay, but it has no concept of time, right? So we talked about how we might want to smooth over time. Okay, so how would you do that? Well, one possible way is to add a temporal smoothing component to factor analysis. Okay, and one way of doing this is a method that some colleagues and I developed called Gaussian process factor analysis, where the idea here is that the Gaussian process part is doing the temporal smoothing, okay, of the low dimensional state. Okay, and so this is good for single trial analyses and also has temporal smoothing in it. Okay.
Sorry.
Yeah.
So, I missed the point, but what is a good point of having temporal smoothing? Is it like, are you going to, like, kind of easy to analyze, or?
Yeah, it helps to denoise, right? So oftentimes, so if you want to study how neural activity is changing over time, say within a trial with, say, a 20 millisecond time bit, if you use factor analysis, what you'll often see is that the points will be sort of jumping around, right? Whereas for the same neural activity, raw neural activity, if you apply Gaussian process factor analysis to it, then what you'll see are sort of smoother trajectories that come out, right? And so basically what the Gaussian process part is doing is saying if 2 points are close together in time, they should also be close together in the low dimensional space, right? So that's sort of a definition of smoothing. Right. Okay. So one of the drawbacks with GPFA is that all the GP does here is to smooth, right? There's no sort of, if you want to try to extract Newton's laws, right, of how this neural activity plays out over time, this GP doesn't give it to you, right? You need some explicit dynamical model, right? And so this is where latent dynamical systems comes in, right? Where rather, so in the low dimensional space, rather than simply smoothing, right, we might define, right, that there is some equation, right, that describes how the low dimensional state changes over time, right? So the simplest version of a latent dynamical system is a latent linear dynamical system, right? And that takes the form of xt equals a xt minus 1, right, plus noise. Okay. So the last sort of broad category of methods is nonlinear methods. Yeah?
So if are these latent dynamical systems?
Uh-huh.
So if you know, for example, that many neurons in sensory cortices tend to adopt their firing rates with time, can you incorporate this kind of rule, for example, in the latent dynamical system?
Yeah.
A rule that governs the dynamics onset and offset responses, for example.
Yeah. So if there are, there's a priori knowledge, right, about what goes on in that brain area during this task, right? You might want to build that in, right? And that way, that helps to constrain what's going on and might help you to denoise, right? So absolutely. So in terms of nonlinear methods, right? So one might ask, right? Why are we assuming a linear low-dimensional space? Right? So in that cartoon that I showed, we had a plane within a 3D space. Why couldn't it be a warped plane? Right? So in principle, yes. I mean, I would guess that these low-dimensional spaces are nonlinear. Okay? But it turns out that there's been a lot of work, right, in machine learning and statistics about how to identify nonlinear low-D spaces, right, from high-dimensional noisy activity. But those methods usually have trouble in settings where there's noise. Okay, so oftentimes in these papers, you see that they work really well on sort of basic demonstrations, and usually they're on noiseless data. But then when the data are noisy, then it's more difficult, right? And so when we're looking at neural activity, typically the data are quite noisy, right? And so it's tricky to use these sorts of nonlinear methods, right? So if you're going to use a nonlinear method with neural activity, I would say use with great care, right? And ask yourself critically whether what you're getting out is something reasonable or actually something the brain is doing rather than the method.
I mean, there's two problems with these methods, right? One is the fact that they don't have good noise models, and the other one is that they're not explicitly meant for time series data. So they assume that you have some underlying latent space that's uniformly sampled, but when you have time-based states, you don't sample uniformly, but you sample these trajectories through your space. So
Right,
there's a
right.
danger that this throws off.
Right, yeah.
With
Yep.
T-SNE counters a nonlinear method.
Yes, indeed. Yep.
That's becoming quite popular recently from
Yep.
what I've seen.
Yep, yep, yep. Yeah, so as with any method, they make some assumptions, right? There are some properties that might need to be satisfied for them to work well, right? So oftentimes, so I'm not sure about T-SNE, but for some of these methods, there's some, like as Jakob was saying, there's some property that ideally would be satisfied. And I don't know off the top of my head what it is for T-SNE, but it would be good to know what that is, and then ask whether the neural activity that you're looking at satisfies that property. Okay. All right, so that's all I have for slides. Okay, so I want to go through this in a way that's sort of adapted to what's most helpful to you. So I'm going to ask you questions along the way, like, are you familiar with something? And then you guys let me know, right? Okay, so who is familiar with PCA? Okay, awesome. Who is familiar with factor analysis? Okay, I see a few hands, but all half raised. Okay, good. Okay, so it turns out, okay, so we had the most votes for GPFA. So I think we'll focus on that. And it turns out that GPFA is only a slight modification on factor analysis. So I think it's first useful to go through some of factor analysis to get some intuition for that. And then we'll add on the GP part, and then you get GPFA. Okay, is that reasonable? Okay, great. Okay, so factor analysis, okay, is based on the following probabilistic model. So okay, so we have Z, okay, which is going to be our latent variable. Okay, so we're going to say this is Gaussian with mean 0 and unit covariance, where Z is in RP. Yeah.
What's the little bar below the Z?
Ah, it means it's a vector. Yeah. Great.
You didn't know that?
This is a stupid question.
Excellent. Yes, please ask if. So this notation, right? I don't know if this is too pedantic, right? So this is saying Z, right, is a random variable, or a vector in this case, random vector, right? That's a Gaussian, right, with mean 0 and covariance, right, which is the identity matrix, which is P by P in this case. Okay. Our observed variable, we're going to call X. Okay, given Z. Okay, it is Gaussian. Okay, with mean C times Z plus D with covariance R, where X is in RQ.
So that's a linear assumption.
Yes. Okay. So what this is saying, right, is, okay, so C, right, is a Q by P matrix, right, where, I should say that in general, Q is greater than P. Okay, so Z is our low dimensional variable, right? X are our high dimensional observations, right? So if you're recording from 100 neurons, right, and you want to extract a 10-dimensional latent space, right, then X is going to be 100-dimensional, and Z is going to be 10-dimensional. Did I say 100? Yeah, 100, 10. Right? Okay, so in that case, C is going to be 100 by 10, tall and skinny matrix. Right? And what this matrix does is it defines the relationship between the low-dimensional space and the high-dimensional space. Right? So another way of thinking about this is the columns of C, right? The span of the columns of C, right, define the low-dimensional space, linear space. Right. Yep.
So, again, because I'm not familiar with this, and maybe some of the students aren't, what justifies the linear assumption?
Right. So.
Could be any polynomial, right?
Yeah. So the reason why we're starting here is because this is, okay, so we start with PCA. That would be like the most basic method, right? Factor analysis is like one step above from there, where we incorporate the idea of observation noise, right? And then we're going to then we can branch off in a couple different directions, right? One direction we can go in is time series models, which is where we're sort of heading down in the next sort of half an hour or so. Another direction we can head down is thinking about nonlinear relationships between X and Z, which we're not going to talk about today. But people have worked on this. I mean, this has been a hot area in statistics and machine learning in the last couple decades.
This is possibly a very technical side point, but one way you can think of factor analysis in PCA is also in terms of ways of parameterizing covariance matrices. While they're defined on data, what they really do in the end is to look at the covariance matrix of all your data and try to express that the n matrix using a lower number of parameters. So you will end up having a number of parameters that is much lower than n squared, but still approximates your n squared matrix.
Yep.
So in that sense, given that it works on the covariance matrix, nonlinear higher order effects are ignored because they don't matter to the covariance.
Yeah. Yeah. So we can make that explicit. So these two distributions, okay, are a full definition of the factor analysis model. Okay. So let me, I guess, before we get back to Jakob's point, let's talk about what these parameters are. So D here, right, is a Q by 1 matrix. Right? You can think about this as the mean activity for each neuron. Okay. R is a Q by Q diagonal covariance matrix. Okay, so it's important here to specify that R is diagonal. Okay, and so each element along the diagonal here you can think about as being the noise variance for each neuron.
The assumption here is that the noise is independent within neurons.
That's right. Yeah, yeah. And so yeah, so this is a really good point, which is every method makes assumptions, right? And whether or not those assumptions are good, we need to. It's an empirical question, right, for what you're trying to do. What type of data do you have, right? So the assumption that factor analysis is making here is that the thing that we want to keep or to extract, right, to characterize in this low-dimensional space are changes in activity that are shared across neurons. Okay? And it's going to discard any aspects of activity that are not shared with other neurons. Okay? So if you look at the activity of one neuron, its activity is going up and down over time or across trials. Okay? And if this fluctuation, or part of this fluctuation, is correlated with at least one other neuron in the recorded population, then it's going to keep that in the low D space. And then any aspect of that variation that is not correlated with the fluctuations of any other neuron in the recorded population, it's going to discard that. And this R captures the part that's discarded. It's saying for each neuron, how much variance did I discard? Okay. So now coming back to Jakob's point, right? So it turns out that this probabilistic model, okay, can be rewritten, okay, only in terms of x. Okay, so x, it turns out, is also Gaussian, okay, with mean d and covariance C transpose plus r. Okay, so it turns out you can write out a few lines to get from here to here. Okay, so we haven't done that here, just in the interest of time, but I'm happy to go through that with you afterwards if you'd like. Okay, but I'm just writing this here to sort of make Jakob's point explicit. Okay, and I think it's illustrative of what factor analysis is doing. Okay, so as Jakob was saying, it's saying that the data that you record, x, right, has some mean d, right? So as I mentioned, you can think about d as being the mean activity of each neuron. So that makes sense here, right? And then what it's doing, factor analysis is doing, is taking the covariance matrix of the activity. So this is a Q by Q matrix. Okay, and it's trying to approximate it with a matrix of this form, okay, which is low rank plus diagonal. Okay, where this low rank part, right, is what we refer to as the shared variance. Okay, and then this diagonal R is the independent variance. Right? So what you can do with this is you can look along the diagonal, right, of this matrix. Okay, so the diagonal, right, of this covariance matrix, right? This is the raw covariance of the neural activity, right? Is just saying, okay, for each neuron, what is its variance? Okay, and what it does is that this partitions each neuron's variance into a component that's shared among neurons and a component that's independent across neurons. Okay, and it's the part that's shared among neurons that we're going to preserve in the low D space. And the part that's independent across neurons, we're going to discard. Yep.
Sorry to keep
No.
interrupting
No, no.
you, right? But naively, I would think that any independent variance would carry more information than a shared variance.
Yeah, what is the
A shared variance, you can kind of express what one neuron is doing in terms of what another is doing, but an independent variance is additional information.
Mm-hmm. Yeah, so what you can do here, right, is so using this decomposition, right, you can partition your raw neural recordings into the part that's shared and the part that's independent across neurons.
But if you now discard the independent part,
Okay, so you can have what's
losing
remaining.
crucial information.
Right.
I'm being very naive here,
Right.
but I'm hoping that for some
Yeah,
people
yeah.
who
So
are
what I
not that
mean-
mathy,
Yeah, so what I mean by discard is it's not being captured by the low D space. Okay, but you can take the residuals.
so your intent is not to capture the entirety of information, but to reduce to a.
Yeah, so the part that's discarded, you can still know exactly what that part is, the part that's not being captured by the low D space. It's the residuals of this model. And if you think that that's important, then you can try to analyze that, right? You can try to correlate it with some, or relate it to some variables. So it's not lost.
Yeah.
Yeah, it's just not captured by the low D space. That's what I meant.
Right. So you have an abundance of information, you reduce it.
You have abundance of activity, let's say.
What
Yeah.
what you're not including in the reduced
Yeah.
description. Yeah. Okay. And
Okay.
I'm just curious because I lectured yesterday, you know, like how mathy people are, you know, like how many people are kind of a little bit lost here. Nobody wants to admit it. How many people are completely confident that they understand everything that's going on here? Also nobody.
Yeah. So do you guys want to ask some questions about? Because I'm acknowledging that I'm skipping steps, right? So if you were to give me this, I wouldn't immediately see this. Yeah. Okay.
How would you choose set and P?
Which?
I mean, you have X given, right? This is
Yeah. Yeah.
And what would be like
Yeah.
feasible choices, or how would you?
Yeah. We haven't gotten to that yet.
Okay.
Yeah.
Are you gonna take us through an example, like with an example vector or an example, like some sort of?
Yeah. Yeah. So I think that's we'll do that in the exercises. I think it's easier because on the board it's a little bit hard to, because these are vectors and matrices, right? I don't want to multiply matrix for you on the board. It'll be pretty boring for you. But yes, we do want to go through examples. It's very important, right? So that's what the exercises are for. Okay. So so now that we've defined the model, what we can ask is, for let's say you have a data set, right? That you want to perform factor analysis on to get to find sort of a representation of the neural activity in this low D space. Okay, what do you need to do for that? Okay, there are several things that need to be done. Okay, so the first thing is we need to fit the parameters of this statistical model to our data. Okay, so the parameters here of this model, so parameters are C, D, and R. Okay. Q, right, is defined by the data, right? And then P is something we'll need to estimate from the data. Okay, we'll talk about how to do that. Okay. So in order to estimate the parameters, okay, the typical way to do it is to use the expectation maximization algorithm. Okay, who has seen or is familiar with the EM algorithm? Raise your hands high. Okay, so in the interest of time, we don't have the time to get into the EM algorithm right now. Right, so usually in my course at home, I go through the EM algorithm, and that itself is 3 hours. So what I'm going to tell you is sort of the intuition behind what it's doing. Okay, so in order to estimate these parameters, okay, the thing that we're going to try to maximize is probability of our data. So I'm going to denote our data as brackets around the x. That means a collection of x's, right? Because we have a bunch of vectors x as the data that we're analyzing. Okay. Under the setting of these parameters, C, D, and R. Okay.
Is the bracket is at multiple trials then?
Yeah, multiple trials or multiple time points.
Mm-hmm.
Right. Okay. So what we're going to want to do. Okay. So for any setting that you choose on these parameters, C, D, and R, And for the data that you observe, right, so this is fixed, right? But we can twiddle these parameters. Okay, so for any setting on these parameters, we're going to get a numerical value for this probability as computed with this. Okay, so what we can ask is how do we twiddle the values on these parameters to make this as large as possible, right? So the reason why we want to do that is because what we're asking here is what is a setting on these model parameters that makes the data that I observed in the real world as likely as possible, right? That is the definition of a good fitting probabilistic model to the training data. Okay, so how do we do that? Well, one way to do it is to try to take derivatives of this directly with respect to D, C, D, and R. Okay, so if you can do that, great. Do it, right? But oftentimes, when you have latent variables in your model, doing that directly is difficult. Okay, and in that setting, typically, if we know what the latent variables are, which we don't, but if we knew what the latent variables were, then estimating the parameters would be easy. Okay, so what the EM algorithm does, okay, is it breaks down this hard problem into a large number of simpler problems. Okay, and so what it does is it iterates. Yep.
I'm confused because the equation you wrote down doesn't have the latent variable anymore.
Yep.
It seems like you should be able to take the derivative
Yeah. So.
without resorting to the latent variable.
Right. Okay. So with factor analysis, there are many different ways to fit factor analysis models, some of which do so more directly, as you're describing, and the other way is using the EM algorithm. So the reason why I'm sort of presenting it in the EM algorithm way is that once we switch up this model a little bit, right, whether we add the Gaussian process part, which we're going to add, or we change this observation noise so that it's no longer Gaussian, for example, then oftentimes doing the direct derivative is difficult, right? So if we have sort of the EM algorithm in mind or in place, we can change any of these aspects of this probabilistic model, and then we turn the crank again on the EM algorithm, because the EM algorithm is completely general, right? You can apply it to any probabilistic model with latent variables, right? So once you know how to use the EM algorithm, you can change the assumptions of your method based on your needs, right, for the analysis you're trying to do. And then you can come up with a way to estimate the parameters of the model and also the latent variables. So it's sort of a more sort of general approach that we're taking here that can be applied to different contexts. Yeah.
Sorry, can you say again which are the latent variables?
Yeah, so Z, right, so this is the latent variable. And then X is the observed variables.
I thought you said that D was akin to the mean.
Yeah.
So then how exactly is it a parameter that isn't just measurable?
Yeah, so it turns out that in this case, if you do the EM algorithm or you take a derivative of this with respect to D, as Christian was suggesting, you get exactly the mean of your data.
Yeah.
Right? So it turns out that what you can do is to just find the mean of your data, just plug that into D, and then only fit C and R here. Right? So there are definitely simplifications that.
But why wouldn't you do that sort of normally since you have the data?
I would, yeah.
Okay.
We would, yeah. So the reason why it's not here is that we don't get to observe Z directly. Right? So if we observe Z directly, then what you want to do is to put Z here. But we don't get to observe Z directly. So what we need to do in this case is to consider all possible values of Z. And so the mathematical way of saying that is we integrate over all possible values of Z. And that's what we did to get this. We integrated over all possible values of Z, to get this distribution, this marginal distribution on x. And so that's what we want to maximize here. So in this case, right, so we have sort of two types of vectors, right, z and x. Right? So we have marginal distributions. This would be probability of z, right? We also have probability of x. Okay, we have joint distributions. Right, so in this case, we have just one joint distribution, which is probability of z and x. Right, and we also have conditional distributions. Okay, we have probability of x given z and probability z given x. Okay, so how do these play a role here? Okay, so probability z, right, is just this. Okay, this is a given in the probabilistic model. Okay, probability x is just this. Okay, the joint distribution, right, we can rewrite the joint distribution. So probability z and x equals probability of z times probability of x given z. Right? So we can get this by multiplying these two Gaussians. Right? So this is the first, we call this the state model. Right? And then the second term here is this observation model. So we can get this joint distribution by putting those two together. Okay, and then the conditional distributions, probability x given z, this is this. Okay. And then probability z given x. Okay, what is probability z given x? Okay, turns out that this is an important distribution when we're doing dimensionality reduction. Okay, so this is saying that we observe some data vector x. Okay, and this is saying what is the probability that different z vectors correspond to the x that we observed. Right? So one way of going from taking a high dimensional data point x and then finding the point in the low dimensional space that corresponds to it is to find the expected value. So the low d projection we often define to be the expected value of z given x. Okay? And this is based on this distribution. Okay, Christian, I'm glad you asked about this because these are all part of like the components of this, right? So now that we've introduced this, we can use these different components together. Okay, so back to this for a moment. Okay, so we want to find the parameters, right? C, D, and R. And we said that one of the ways to find the parameters is using the EM algorithm. Okay, and we want to turn what is potentially a difficult problem into a sequence of easier problems. Okay, so what we do with the EM algorithm is we have a so-called E step and a so-called M step. Okay, and then we keep performing. So we do an E step and then another, and then an M step, and then another E step, and then another M step, and then another E step, and then another M step until convergence. Okay? And at each step along the way, after you do one E step and one M step, you can guarantee that this thing that you want to maximize goes up. Yep.
I'm lost again. What are E steps and M steps?
Right. So those are the two key components of the EM algorithm. Okay? So we want to maximize this, okay, with respect to the parameters.
That's the E step.
Now, that's the overall objective. Okay. So this is the overall objective. Okay. So, as I mentioned, right, if you want to do this by taking derivatives and you're able to, then that's great. But if not, right, you might, and there are latent variables in the model, you might consider using the EM algorithm. Okay, so for the EM algorithm, there are two key parts. Okay, so one is the E step, and one is the M step. Okay. So it turns out, okay, that there's a general recipe for the EM algorithm that you can apply to any probabilistic model with latent variables. Okay. And what that recipe says is that during the E step, what we want to do is to compute the probability of the latent variables given the observed variables. Okay, so in this case, we want to compute probability of Z given X, okay, which is this. Okay, and then without getting into details of the M step, well, here what we want to do is to update the parameters. In this case, it's C, D, and R. Using the distribution found in the previous E step. Okay, so the idea here, right, is that when you're faced with a difficult problem like this, and the reason why it's difficult to solve directly is because you have latent variables, right? Oftentimes in these cases, if you knew what the value of the latent variables are, maximizing this would be easy. Okay, so what the EM algorithm does is it says, okay, during the E step, what I'm going to do is to take my best guess as to what the latent variables are. Okay, and that best guess is based on the observed data, okay, and the most recent or the most current estimate of the parameters, in this case C, D, and R. Okay, and then during the M step, what you do is you say, okay, well that tells me sort of roughly what the latent variable value is for Z. I'm going to use that in the M step to update the parameters. Right? And then you take those updated parameters, and then you do the next E step, right, where you now can compute this distribution again with a new set of parameters. Right? And you go back and forth, right? And it turns out that doing this procedure, you're guaranteed to converge at a local optimum. Okay? Meaning that after each E-M step combination, you're guaranteed to increase this overall objective. Yep.
I don't know, I know this is not the same as gradient descending. For example, I can see in gradient descent how no matter if the gradient or positive or negative, you always go towards the minimum. But I don't understand here why if you just guess randomly and keep updating, you would converge towards the maximum of P. Why is that?
Right. So here we're not guessing randomly, right? So that's the key, right? So what we do is we say, okay, we need to start off with a set of parameters C, D, and R. Okay, and so one way of doing that is to initialize with a random setting on the C, D, and R. Okay, and then we say, okay, well, given those settings on the parameters, what is our best estimate of the latent variable Z? And then using that best estimate of the latent variable Z, we make our best estimate of the parameters. And using that best estimate of the parameters, we then update our best estimate of the latent variables. Right? So yes, there can be randomization when you first initialize these parameters. But after that, there's nothing random to it. Right? We're trying to make our best estimates of the latent variables and the parameters. And so without getting into the math, which will take a couple hours, it's a little, so it turns out that you have guarantees, right? As I said, so after each E and M step, you're guaranteed that this increases. But it would take us a little bit of time to actually show or prove that it does that. So what we're going to do in the exercises this afternoon is you can implement your own EM algorithm, or you can use code for factor analysis that I provided, and then you can see that indeed, after each EM iteration, that this probability increases.
For that likelihood, you have
Yeah.
you have a general formula, you have to propose something, or
Yeah, this one?
yeah.
Yeah, so this is just this, right? So this thing has to be computed based on the probabilistic model that you want to fit to your data.
Okay.
And in this case, the probabilistic model that we want to fit, or since we're talking about factor analysis, is this one.
Okay.
Yeah. So it turns out that the fact that these distributions are Gaussian, and as a result, this one's a Gaussian, it makes the math nice, meaning you can compute all the necessary quantities required for the EM algorithm exactly. Okay, so if you switch out this observation model, and let's say it's no longer Gaussian, but Poisson, right? Then you might not be able to compute everything here exactly as prescribed by the recipe of the EM algorithm. And so if we can't compute those quantities exactly, then we're no longer guaranteed that this thing increases after each EM iteration. But oftentimes, hold on one sec. But oftentimes, if you can get really close to that value, you have a way of approximating it, then you might still be able to increase this value considerably without the guarantee. So what this guarantees is that you converge to a local optimum.
Yes.
So this implies that where you end up could depend on where you start. So if you start in a bad place, you can end up in a bad place. So this is sort of a standard issue with many statistical and machine learning algorithms, right? And so one way of dealing with this is to initialize at different locations and see whether they end up all in the same place. And if not, take the best of those, of where you end up. Right. Jakob, were you going to say
I
something?
just wanted to add that if you're in a non-Gaussian world, there's an extension of the end, that's called a variation of the end, that still, it doesn't guarantee to optimize the lower likelihood at every step, but a lower bound. So there's a way to extend this to non-Gaussian families that still gives you guarantee of convergence, but not that you necessarily maximize it.
So one reason is because X might represent spike counts in one big bin, where you only have one bin on each trial. Right? So there, you still have the sequence of trials, but if what you're interested in studying is sort of the structure of how the neural activity changes across neurons, then one might want to use something like this.
Sorry, if you're just looking at a single trial.
Right. So if X represents a spike count for each neuron on one trial in one large bin. right? Yeah. Another reason one might want to use this is if, okay, so one thing to remember is that every statistical method makes assumptions, right? And so the different time series methods that we could use here, right, like Gaussian process factor analysis or latent linear dynamical system, each of those makes assumptions about what the time series is and can influence the results. And so we need to assess critically whether those assumptions are appropriate for what we're doing, right? And so without sort of a strong reason to say like one of those different types of time series models is appropriate, and if we're not actually looking at trajectories in this low dimensional space, and if instead we want to compute like basic metrics, like what is the dimensionality of the data, right, based on a linear method like this, then one might want to use this, right? Okay, and I'm happy to help or chat with you about any of this, so feel free to flag me down in the afternoon.